Hi everyone, welcome to the enzyme lecture. So we are going to review some old things about enzymes that you should remember from honors biology and perhaps some new things as well. Chapter 6 has a lot of material in it. For now, we're only talking about sections 4 and 5 of this chapter. So if you are an all remote student, you have access to a full printout of the notes with everything filled in. You're welcome to just print those out and keep them, or just save them to your virtual binder. If you don't have a printer, you're welcome to take notes on a separate sheet of paper as I go along. However you want to do it, it's up to you. If you are a student who's been in class, you would have received a copy of these notes here, the PowerPoint slides with words missing on the slides for you to fill in. I'm going to be drawing or writing some things on some of the slides as we go through today, so you're welcome to write that down again on just like a separate sheet of paper. If you're an all-remote student, again, you can print them out if you'd like, or just follow along and listen. It's really up to you what you choose to do, and this lecture will always be available to you. All right, so let's get started here. An enzyme is a catalyst. So you might remember from chemistry, or you might remember from honors biology, that a catalyst is something that speeds up a chemical reaction. So enzymes are proteins that speed up chemical reactions. So let's talk about an example of how that would work. Let's say, for example, that I have a beaker of sugar. So here's my beaker, and I set it on the demo desk in my classroom. Okay, so here's my sugar. We'll say that it's sucrose. Okay, so I leave my sucrose on the demo desk in my classroom, and I leave it there for hundreds of years. And somebody comes back and checks on it. Over time, that sucrose is going to break down. It's going to take a really, really long time for it to happen naturally, though. It will break down into its two monosaccharides, which are what? Do you know? Right? Glucose and fructose. All right, but with the help of an enzyme, with the help of the enzyme sucrase, remember from last year that enzymes usually end in ase. With the help of the enzyme sucrase, that reaction is going to take place in mere seconds. It's going to break down very quickly by the addition of this protein catalyst, aka an enzyme, to the chemical reaction to break down this disaccharide into its monosaccharides very quickly. So here's an example of that chemical reaction that I just talked about. In this particular case, the enzyme is sucrase, and it's always helpful when the enzyme and the thing that it breaks down have the same or similar names. Sucrose, remember that sugars usually end in ose, and enzymes usually end in ase. So sucrase breaks down sucrose. And that this chemical reaction, again, is going to take place in mere seconds versus the hundreds of thousands of years, like my example here with the beaker. All right, so all chemical reactions involve bonds breaking and reforming. Molecules are put into unstable states when a lot of energy is absorbed from their surroundings. So when new products form or new bonds form, they release heat and energy, and molecules go to a stable state. So we'll look at an example of that on the next slide, but you should have some knowledge of that from your chemistry classes. So here's an example here. Enzymes, what they do ultimately is lower the activation energy that's required for a chemical reaction, which I believe is what, yes, this slide talks about. Okay, so the initial investment of energy for starting a reaction is called the activation energy, which was the word we saw on the last slide. So the energy that gets absorbed from the surroundings causes more collisions of molecules, more breaking of bonds. So here in our chemical reaction, we have the reactants, right? They're always on the left-hand side of my chemical reaction. My reactants are A bonded to B and C bonded to D. Now, these molecules or reactants are going to absorb energy from their surroundings, that activation energy. And what happens is when they absorb energy, up here they're in an unstable state. Okay, so that happens again because they absorbed energy from their surroundings. So that could be things like heat energy, right? It's some example that probably something you learned in chemistry last year, when these molecules absorb energy. It causes their bonds to eventually break when they get so unstable, and then you get new bonds that form once those bonds break. Where's my cursor here? Okay, so once so much energy is absorbed, the bonds break, and then new bonds form. And we see that down here in the picture, right? Now we have different products than we had before. A is no longer bonded to B, and C is no longer bonded to D. We got new bonds that formed, and when that happens, you should remember this from chemistry, energy is released when new bonds form. All right, so you should have learned that or heard that before in chemistry class. But when that happens now, you have new molecules that have formed once those bonds were broken from the original reactants, and energy is released from that chemical reaction. So what does that all have to do with enzymes? Enzymes catalyze reactions by lowering the amount of activation energy, or the EA that we see here, okay? They lower that amount of energy that's needed for a reaction, and it makes the molecules go into that unstable state that we saw on the last slide here, where the bonds were wiggly, right? That means unstable, where that happens a lot quicker with, or a lot faster, that unstable state, with the help of an enzyme. That's going to make the whole thing go a lot faster. All right, so that's essentially what they do. So let's talk about some vocab words from, hopefully you remember, honors biology, I hope, right? So the first one on here is substrate. You should remember the word substrate. A substrate is the thing that binds to the enzyme, all right? So a substrate is a thing that binds to the enzyme. It's all, sometimes you'll see it called the reactant. All right. The enzyme substrate complex is the combination of the substrate bound to the active site of the enzyme and the enzyme itself. So it's the whole combination of both. If you were in my class last year, we cut out pictures of an enzyme and a substrate, and you stuck them together, and then you had to label them on your paper as the enzyme substrate complex. You might remember doing something like that. So it looked like this if you were in my class last year. Here's the enzyme, and then here is the substrate. When you combine those things together, you take your enzyme and your substrate, the combination of those things is the enzyme substrate complex. So a complex in biology is something that is the combination of multiple proteins or multiple molecules. So in this case here, when the substrate is bound to the enzyme and it's the combination of all those things, you have your enzyme substrate complex. And the other important term on here, or something to remember, is the active site. You should remember that hopefully from, again, honors biology last year. So the active site is like the groove or the spot on the enzyme where the substrate is going to bind. All right, so hopefully you remember all those things. And you should remember also from last year, enzyme specificity. You may have heard myself or Mr. Rosa call this a lock and key. Do you remember that from last year? Enzymes and substrates are specific to one another. Certain substrates only fit certain enzymes, just like only certain keys fit into certain locks. They are a biological fit. They are designed to fit together. And we call that induced fit. So when a substrate enters the active site, the enzyme changes its shape slightly due to the interactions between the substrate's chemical groups and the R groups of the active site, and this changes the shape slightly like a handshake. So it's not, so although it's a lock and key like we talk about in honors biology, it's more kind of like if you think about a handshake. When you go to shake somebody's hand, right, you extend your hand out, but then you kind of change the shape of your hand a little bit to mold around the other person's hand. Of course, we can't shake hands now, right, in 2020, not in 2020. But you can think of a handshake when you think of the induced fit. It's when your enzyme is slightly changing its shape in order for that substrate to fit into it. So let me show you a picture of what that actually looks like. So here's our enzyme here, okay? And this is our substrate. So you can see the shape of the enzyme before the substrate binds. Substrate binds to the active site of the enzyme, and look, now it's kind of like a different shape over here, right? It's kind of more clenched, like your hand would be if you were shaking somebody else's hand. So when this happens, the bonds that are all here around this substrate are all forming weak reactions or weak attractions to the chemical or the molecules that are inside of that substrate. So you see all these little circles here, all these little blue circles on the screen. What do you think they are? Enzymes are proteins, right? Think back to your biological molecules. Yeah, so these are all amino acids, right? If an enzyme is a protein, it's made up of hundreds of amino acids all linked together. That's what all those little circles are in the picture. All right, so you should hopefully remember that. Okay, so let's talk about how enzymes lower that activation energy. Enzymes use a variety of mechanisms to lower activation energy. One of the first things they do is that they provide a template for the substrates to come together or bind. So one of the things that they will actually do is kind of orient the substrates in the right way, meaning they will kind of mold them or move them as they do that induced fit to get that substrate to fit better into the active site. Enzymes can stretch the substrate to a transitional state where the bonds become unstable. So they'll do that by actually like straining the molecules, breaking those bonds or causing them to become unstable. You can also have a situation where the enzyme provides a favorable microenvironment. So what does that mean? Well, on the last slide I just showed you with the enzyme here and all the amino acids together, you should remember from our amino acid structure that they have a central carbon, right? A carboxyl group, an amine group, and then they have an R group, right? And I don't know how far, depending on where your class is at this point in the week, all these R groups are different on all the amino acids, and they all have different chemical properties. So depending on the chemical properties of those R groups, that can be attractive to certain substrates. So you can have some R groups that are acidic. You can have some that are basic. You can have some that are polar. You can have some that are non-polar. So it really just depends what kind of amino acids make up that enzyme, and the certain substrates are attracted to certain enzyme areas or the R groups that are pointed out, and here I'll show you here, pointed out are kind of exposed, if you will. So all in here, this area here, these would be R groups facing out with different chemical properties to attract this particular substrate. I'll give you an example. In the cell membrane, if you remember the cell membrane, yes, with all the phospholipids, okay, remember that? Water, if you remember from Honors Bio, doesn't like to just go right through the phospholipid bilayer. It actually goes through these channels that are called aquaporins, and we'll talk about this more in the next unit. But an aquaporin is a protein, and in that protein, there are amino acids with R groups that are inside the channel of that protein that are all polar. And why do you think that is? Well, water is a polar molecule, right? Like dissolves like. So water would go right through that aquaporin inside the cell because it's attracted to the R groups that are facing inside of that channel inside the cell membrane. Similar idea with enzymes and substrates, right? But we'll talk about that more when we get to the next unit. But that's what's kind of happening here with how the enzyme is able to attract that certain substrate to that area. And then covalently bonding to the substrate, that makes sense, right? It forms those chemical bonds. Okay, so if we're looking at this picture here, the substrates enter the active site, the enzyme changes shape a little bit. That's that induced fit that we just talked about. Once that happens, once the two come together, the substrate is held together by weak bonds, usually hydrogen bonds or ionic bonds in the active site. The substrates are then converted to products. The products will be released from the enzyme, and then the active site is available to be used again. You should remember from last year that you never run out of enzyme. You never run out of enzyme. Right? Enzymes can be used over and over and over again. It's the substrates that can vary in their concentration or how much you have of them. And when we do our virtual enzyme lab next week, you'll see more about that. All right, you should remember these things from honors biology as well. If you don't, that's all right. But there are certain things that can change an enzyme's ability to function. Temperature is a big one, and pH is a big one. So the temperature can alter the shape of an enzyme, as can the pH of the environment around that enzyme, and it can affect its ability to function. So next week when we do our virtual enzyme lab, we're going to be manipulating the pH, but these things can change the ability of the enzyme to function. So we're almost halfway done here, okay? So what happens actually when different temperatures and different pHs are introduced to enzymes? Well, proteins are pretty sensitive to their external environments, especially temperature. That can increase the rate of the enzyme's reaction or speed up that thermal agitation of the enzyme. It's going to disrupt hydrogen bonds within the molecule, ionic bonds, and other weak interactions that stabilize that active shape of the enzyme, and it will eventually start to denature them. The same is true for pH. So I just said the word denature. You should remember what that means from honors biology, but if you don't, it means to change shape. So changes in thermal energy, changes in the pH of the environment are going to change the shape of the enzyme, therein affecting the ability of the substrate to bind to those enzymes. So here's two examples here of environmental factors affecting enzyme activity. The top one is temperature and the bottom one is pH. So it's different for different enzymes. So here's an example in the top left-hand, or the top here with temperature. Sorry, where's my mouse? Okay. So here's an enzyme that is in the human stomach or inside of a human, and the x-axis down here is in Celsius. So about 36 degrees Celsius is about 98 degrees Fahrenheit for humans. So we know that that's our optimal body temperature. When you expose or when you have enzymes in your body, they're going to be best reacting or best acting at the body temperature of the organism they live in. Now, if you look at another example of an enzyme, here's an enzyme that is in bacteria that is heat tolerant at 77 degrees Celsius. That's very hot. If you put that same enzyme into a human, look, the graph stops, right? The reactivity or the rate of the reaction comes to a screeching halt because what has happened here in these areas where there is sloping down, that enzyme is denaturing. It's changing shape, so that the active site is not easily able to bind to the substrate anymore. So wherever you see a downward slope, that's that denaturing that's happening here. Same thing with pH. So here is an enzyme that is in the human stomach again. The human stomach is very acidic. You should remember the pH scale. Acids are on this side, bases are on this side. The human stomach is very acidic. We have that hydrochloric acid that's in our stomachs. So, give you an example here. Lemon juice has a pH around 3 or 4. And if you've ever gotten lemon juice right in a cut or something like that, you know it stings and burns. I think it might actually be more around a 4. Somebody can fact check me on that, I'm sure. But anyway, so your pH of your stomach is very acidic. So you're going to have a very acidic environment for enzymes that function in your stomach. If you took that very same enzyme and put it into your intestines are nowhere near as acidic as your stomach because they don't perform the same function as your stomach. Your stomach is for digestion, so enzymes that are in the stomach are going to be breaking things down, whereas enzymes that are in the small intestine are not. They're more for absorption, so they don't need to function in as much of an acidic environment. So you can see they function at a different pH, a more basic pH of 8. As you look at the graph for the stomach pH and the stomach enzyme. Once you get past 3.5, that enzyme is no longer functioning. It has no activity whatsoever because it's not able to function in a different environment. And that's basically the point of those graphs. Okay, and here they are again in bigger form. All right. So many enzymes require what they call non-protein helpers or cofactors. So they also can require these things called coenzymes. They can be permanent or they can bind loosely to the enzyme and kind of help it get through a chemical reaction. Some good examples of cofactors would be things like metals like zinc, iron, copper. All right, so these metals can bind to an enzyme and help it perform a chemical reaction. And as I said a minute ago, they can bind permanently to the enzyme structure, or they can bind temporarily and then come off that enzyme when the chemical reaction is done. Some examples of coenzymes, spelled that wrong, would be things like, and you'll read this in the book, of course, would be things like vitamins. All right, so these again are non-protein helpers, so things that are binding to the enzyme to help it work or not. And then certain chemicals inhibit the action of specific enzymes. So there are things like toxins or poisons that can inhibit an enzyme or prevent it from working. So examples like nerve gas, right? That shuts down your whole nervous system. Well, how does that actually work? Anything that's like that, that's like a toxin or a poison, it can be a competitive inhibitor or it can be a non-competitive inhibitor. We'll look at pictures of them in a second here to explain more of what they are, okay? So competitive or non-competitive inhibitors. So here's a situation with a normal enzyme. The substrate is able to successfully bind to the enzyme. It would release the products and the chemical reaction would take place. Now you have competitive inhibitors. So imagine this is, again, an example of something like this would be like a toxin or a poison. All right, something that is going to have the same shape of the substrate that can bind to the active site of the enzyme, and it will prevent the substrate from binding to that enzyme. So now the substrate's like, hey, you're in my way, I can't fit, right? So now this other molecule has competitively stopped or fought for the space in the active site and prevented the substrate from binding to it. All right, so this is something that not a competitive inhibitor is something that mimics the shape of the active site, or I'm sorry, of the substrate. Mimics the shape of the substrate, and then it's able to fit to that active site. All right, and then there are also other kinds of molecules similar to your, again, toxins, poisons, things like that, non-competitive inhibitors. So non-competitive inhibitors, they bind to another space that's on the enzyme. Sometimes it's called an allosteric site. When you watch Mr. Anderson at some point, either this week or next, you'll hear him talk about that. But they're going to bind to another spot on the enzyme, and when that happens, it changes the shape of the active site. So now the substrate is no longer able to bind to that enzyme because this other molecule has bound to another site on that enzyme, again, sometimes called an allosteric site, but this has now changed the shape of the active site, and now the substrate's not able to bind there anymore. All right. So in every chapter of this book, of our book, there is a section that ties back to evolution. So why? Because remember, evolution is one of the big ideas of AP biology. It's 25% of the class. So enzymes being proteins, we know that proteins come from what? You should know, right? They're coded by your DNA, yes? And then we got mRNA, right? In the middle here, mRNA, tRNA. You should remember that from last year, okay? I guess this arrow should technically go the other way. That's my fault, okay? So your DNA codes for your proteins, and your proteins depend ultimately on what the code is of your DNA. So you could have a change to your DNA or a mutation. You should remember that word from last year, mutation. So if a mutation occurs and it changes the letters in your DNA, that's going to change the shape of the protein. That's going to maybe change the shape of the active site that's created once those amino acids are coded for. So when you have something like that that occurs, it could make the reaction more favorable, meaning the substrate could bind faster to the active site, or it might not bind as fast and it might have, a detrimental effect. So over time and evolution, when you've had mutations in proteins, that can cause for, again, sometimes a situation where the active site now is able to better bind to that substrate and you might have quicker chemical reactions. All right. And then the last section of this chapter is pretty brief. We need to be able to turn enzymes off and turn them on at various times. Why do we need to do that? Because there are times when you don't want to be making products that you don't need. Making products that you don't need. So, for example, if there is no sucrose in your system, then you don't need sucrase to be working or being made. All right, why? Because, well, that's a waste of ATP. You should remember that from honors biology. Waste of ATP. You don't want to be wasting cellular energy making a product or having an enzyme working when there's nothing for it to break down, right? That would make sense. We won't want to waste that precious cellular energy. So enzymes that are allosterically regulated have two subunits and two active sites. A protein's function at one site is affected by the binding of a molecule at a separate site for inhibition or stimulation, meaning that you could potentially turn on an enzyme or stimulate it like we have up here, or you could inhibit an enzyme by shutting it off. And there are other molecules that play into this. So inhibitor and or activator molecules bind to an allosteric site on the enzyme and help to turn it on or turn it off. So there are active forms of enzymes and there are inactive forms of enzymes. We'll look at a picture here to better explain all of this. Okay, so on the left-hand side of the screen here, we have an activator that stabilizes the enzyme in an active form, and an allosteric inhibitor that, again, stabilizes the enzyme in an active form. So this, again, is just another molecule that can bind to the enzyme to help it stay activated, help it stay on, help it to function. So with my example of sucrase, if we want sucrase on and working, this doesn't really happen with sucrase, but if we wanted it to stay on and stay working, an activator molecule would bind to it, or an inhibitor would bind to it in order to keep it stabilized, or keep it off, or keep it on, depending on what was called for, what was needed. All right. So in the bottom picture, sorry, that was an inhibitor, not an activator. So similar idea. It's keeping the enzyme off when it doesn't need to be on. And then on the other side of that, you have this idea called cooperativity. So what does it mean to cooperate, right? It means to work together. So in cooperativity in enzymes, you have the binding of one substrate molecule to an active site of one subunit, and it locks all the subunits into an active form of the enzyme. So again, you have an inactive version of the enzyme in the picture here. Once this other molecule, they're calling it a substrate, binds to the enzyme, it will stabilize it and help it to stay on and function. So again, these are just ways of regulating enzymes, ways of turning them on and turning them off. Throughout the year, as we look at different chemical reactions like photosynthesis, right, cellular respiration, we're going to look at enzymes, and we'll see cases where they are turned on and when they are turned off. So for now, this is just kind of something to keep in mind that this can happen. I mentioned cooperativity on that last slide there. There was a picture of it. So cooperativity is when a substrate molecule can bind to an enzyme to increase its activity by amplifying the response. So I'm going to give you an example of what that, your book talks about this example too. So an example would be hemoglobin. Do you remember what hemoglobin is from last year? You thinking about it? So this is the protein in your blood. Helps to carry oxygen. Should remember that, hopefully. All right. So there are studies that have been done with hemoglobin, and it's shown that the binding of an oxygen to one hemoglobin protein or one hemoglobin molecule creates an affinity or an attraction for other oxygens to bind to that hemoglobin in other sites, meaning that the oxygen kind of gets the party started, if you will, and then more oxygens want to come to that same protein and bind there, fill up those remaining sites. So again, that is kind of that idea of working together with the oxygen and the hemoglobin. All right, so that is it for our enzyme notes. You can start working on the reading guide, whatever you need to do. All right, and please let me know if you have any questions.